Okay, welcome to Year 8 Science Online with Henny van Vieren.
Okay, hello again, guys. Welcome to this second online um, lesson that we're going to do today, and we are today going to look at food tests, and specifically the test that we can do to see if there's protein in food, or if there is sugar in food, or a combination of the above, or fats, and so forth. I really wish we could do this experiment practically. It would have made a big difference to your learning, but now unfortunately you just need to remember it. Okay, so let's get going. Okay, so food tests. If we did run the experiment, it would turn out something like this. This is called the Benedict test for sugars. So your specific learning outcomes for today is, for excellence, you can outline the procedures and the results for each test. For merit, I can outline the procedures for each stage and I can describe positive and negative results for each. And then for achieve, I can safely carry out the Benedicts test and the iodine test and the test for lipids. I can describe positive results for each test. Okay, so the Benedicts test. This is the test for sugars. Now remember, we talked about sugars as carbohydrates, and we said that we can have complex sugars, which are long chains of sugar. Remember, sugar can be in a shape of a what do we call it? A hexagon. What was that? In a hexagonal shape? No. I don't like what it's doing here. Undo. Okay, it can be in the shape of a hexagon, or it can be in the shape um, of a pentagon, and consequently they will all line up and form like a long chain, or you can almost think about it as train cars behind each other. And some of them will have branches on that, and those are your more complex sugars. Okay, so what you had to do, you take the Benedict's solution, which is a specific solution that is made up already and had given to you, and you would have added about 2 centimeters cubed, also known as milliliters, of this sample solution to a test tube. So we would have given you different liquids, and in these liquids there might have been sugar or not. Then you add the same amount of Benedicts to that, and then you swirl and swirl to mix up the whole thing. Vortex is just, we don't have a vortex at school, but you kind of make a vortex like you all did in the bottles. Then leave the test tube in boiling water for about 5 minutes, and then the color will change. And when the color stops changing, you know that it's ready to have a look. You look at the color changes during that time, as well as the final color. So what we saw when they came out is that if you had no sugar at all, in other words, if this was a solution with no sugar at all, it would have had a blue color. So if you did this with water, it would have ended up looking like that. Then something with very little amounts of sugar is going to show up as green. There's a real life version. When you have a medium amount of sugar, or a low amount of sugar rather, it turns a lighter orange, almost yellowy, because, yeah, almost, sorry, not because, but almost yellowy orangey. And then moderate, as you can see here, it kind of looks like nice orange carrot color. And then red gave you a um, high amount of sugar contained. So this would have been something like a energy drink or a Coca-Cola or something like that. Okay, so the main thing you need to remember here is that when we have blue, there is no sugar. Green and yellow. Green says a little bit, yellow says a little bit more, orange says even more, and then the closer you get to red, the higher the sugar content. The next one is the iodine test for starch. Now, iodine comes in a liquid form, and you will find it if you've ever had betadine ointment. It's like a brown ointment that we sometimes put on your wounds. It contains a lot of iodine in it, and it's really good at also killing bacteria and so forth. But when it comes in contact with starch, it turns black, completely black. But you can see when you look at the color in this dropper, it is a brownish color, and then it turns completely black when in contact with starch. So if you put this on paper, there's starch on paper, your paper will turn black. Um, Potatoes, obviously, um, complex, well, like pastas and so forth, also have starches in them. Carrots have starch in it. Starch is what a lot of plants use to store their glucose. So they turn them into these long chains, like I said to you earlier, um, and those are long chains of glucose become something that is known as starch. So you simply take your iodine, you drop it on the sample that we would have given you, and if it turns black, then you know there is starch in there. Okay, so that's the iodine test for starch. The next test is the Biuret test for protein. Okay, the Biuret test for protein. So you start off with the reagent or the liquid that is called the Biuret test. It's a specific chemical that we would have given you, and then you add it to your various different mixtures. Now, when you look at it, this is a nice scale. So the darker the blue, the closer you get to purple, the more protein you have in there. So if you, for instance, look at something like egg white has got a lot of protein in it. That would have turned out to be purple like that. But this one is a negative because that's just the color of water. But as it gets more and more blue, it means, and towards purple, it means there's more and more protein present. Okay, so what foods have a lot of protein in them? We know that milk does. We know that I just mentioned eggs, meat, and so forth. Plants and grains also have protein, but they are not complete proteins that we can use as humans, so we need to eat combinations of them. Just like when I showed you in that Bill Nye video, he talked about having to eat different rice and beans together and so forth. And then lastly is the lipid test. Can you remember what a lipid is? A lipid is a fat. So fats and oils, they are called lipids. So what we would have done is we would have given you 95% ethanol. Now ethanol, you will know, is in all your hand sanitizer and so forth. So it's an alcohol. Very strong, highly concentrated solution of alcohol. You then pour it into some distilled water, but you put your sample into this distilled water. If you have this cloudy formation forming, that is actually the fat that is starting to dissolve within this ethanol, forming this emulsion. And then you have this cloudy forming, that's a positive result, and a negative result will simply be clear. And that is the end of today's lesson. So just to recap, we've got the Benedicts test for sugars, and blue means nothing, and the closer we go to red, the higher the sugar concentration. Starch is black when there's iodine, turns black when it comes in contact with starch. The Biuret test for protein goes from a light color, water color, in no or very light blue, to almost a purple color, very dark blue color, the more protein you have. And lastly, the emulsion test for lipids is where the ethanol goes in, you add it to your sample, and if you have a cloudy formation, it means that an emulsion has formed, which means there is a lipid present. Okay, thank you for your attention. There will be some work on the OLE for you to follow up on this. Have a great day and stay safe out there.